Dr. Hansen here. I hope that working through that preview glycolysis video gave you some insight into how to think about enzyme nomenclature and regulation. I do not expect you to remember every enzyme name. I don't expect you to be able to write down the entire sequence of reactions. But if you're given an enzyme name, I hope you could interpret it some to understand the substrates and products, what type of reaction occurred, especially for the enzyme names which are used over and over and over for so many medically relevant conditions, such as kinase, phosphatase, dehydrogenase, isomerase. This all goes back to the pre-matriculation video on nomenclature. Likewise with this talk, I hope you can use a similar approach. Try to interpret, try to understand. Certainly I expect that you will have spent more time on the glycolysis preview video than you will on the gluconeogenesis one. Hopefully you do have some feel for how glycolysis works, because glycolysis is the oxidation of glucose down to pyruvate to make ATP, and gluconeogenesis is the buildup from pyruvate to glucose in order to regenerate glucose, and it costs a bunch of ATP. Accordingly, many of the enzymes are shared, and the language which describes both processes can be understood similarly. Accordingly, I have the same review learning objectives for the gluconeogenesis talk as for glycolysis, and even these new learning objectives on gluconeogenesis parallel the learning objectives for glycolysis, in that they are focused on enzyme nomenclature, regulation, the DAM model, and on energetics. Does it go up or down in energy? Also in parallel to the glycolysis presentation, I want to frame gluconeogenesis in the context of the central metabolic pathway diagram from the textbook. At the top, I'm showing in blue glucose to pyruvate glycolysis. In the middle in purple, I'm showing the conversion of pyruvate to acetyl coenzyme A, which feeds into the citric acid cycle, the circle in the middle in black, and ultimately electrons fall into the electron transport chain in oxidative phosphorylation. My plan is to include the central metabolic pathway figure in almost every talk I give, so that you will be able to map in your mind how the given metabolic pathway that I'm talking about in one talk fits in with the other metabolic pathways. Another way of thinking about how to fit together the different metabolic pathways is in the context of a figure of the cell. This is a learning objective because it gives a lot of insight into how these different pathways work. Specifically, the glycolysis we described in the previous video all occurs in the cytosol down to the production of pyruvate. Pyruvate, the oxidized product of glycolysis, gets pumped into the mitochondrial matrix, where it can enter the citric acid cycle by being oxidized to carbon dioxide in order to acetylate coenzyme A. Acetyl coenzyme A enters the citric acid cycle, which produces a lot of NADH from NAD+, which is high, which is low in energy. That all occurs in the matrix. NADH then binds to transmembrane complexes of the electron transport chain and dumps those high energy electrons into those complexes to fall down the electron transport chain and ultimately to drive oxidative phosphorylation to make ATP. And since these complexes are all transmembrane, and since these complexes interact with substrates both in the matrix and in the inner mitochondrial space, oxidative phosphorylation is best described as taking place in the inner mitochondrial membrane. And this overall sequence is absolutely a learning objective. Of course, I'll be covering the citric acid cycle and oxidative phosphorylation during the activity in Cougar, and right now I want to go back and look at glycolysis and gluconeogenesis. I mentioned that glycolysis oxidizes glucose to pyruvate. How many ATP do you get from that? It's a learning objective. You get 2. Right now I want to look at the numbers. So we know that the hexose phase is the investment stage. You're putting money in. How much ATP does the hexose stage cost? Well, 1, 2. You burn 2 ATP. You're in the hole. You've borrowed money to start your business. So now let's see if we can recoup our investment and if we can make a profit. And that'll occur in the trio stage. So let's see. We have 2 steps named for the reverse direction. I hope you know these enzymes from the glycolysis talk: phosphoglycerate kinase and pyruvate kinase. They're the only kinases in the trio steps. And I'm saying, wait a minute. There's only 2 ATP in the trio steps? Well, we spent 2 AP in the hexoses steps. We're not going to make any profit. Of course, there's a flaw with this reasoning. How do we get a profit? Please pause and answer. It's pretty important to understand. Well, of course, there's 2 trioses for every 1 hexose. So in glycolysis, to prepare the hexoses, you do burn 2 ATP. However, before you recover ATP, the hexose is cleaved into trioses, so there are 2 trioses per hexose. Then, oxidation of each of those trioses produces 2 ATP each, making a total of 4 AT produced, 2 ATP invested to give a net of 2 ATP gained. And I'll say, you know, that's okay, but it's not real impressive. What is going to be impressive is when we take that 1 glucose molecule and go through glycolysis, then go through the citric acid cycle, then go through oxidative phosphorylation to oxidize it all the way down to carbon dioxide and water. In that case, we will get a total of around 30 ATP. And you'll notice I put around or approximately 30. That's because you can't calculate exactly. Theoretically, you can get 38 ATP. You don't need to know that number. There is leakage in the mitochondria, and it also depends on the pathway you follow. So, about 30 ATP. What you absolutely do need to know is that in glycolysis, meaning anaerobic glycolysis, you specifically produce a net of 2 ATP and 2 NADH per glucose molecule. And then in general, when you go all the way through aerobic metabolism, you get about 30. Or in other words, in most of the cells of your body right now, 95% of the ATP were produced by oxidative phosphorylation, and only about 5% were produced directly by glycolysis. In my previous video on glycolysis, I pointed out that most of the steps are reversible, as indicated by these blue double-tipped arrows. Only 3 of the glycolytic enzymes catalyze irreversible steps, shown in red, and they're irreversible because they represent a big drop in energy. These 3 irreversible steps are, of course, also the regulatory steps. For example, hexokinase in phosphorylating glucose to make glucose 6-phosphate is irreversible and regulatory. That is, glucose 6-phosphate is a substrate of glycolysis, so as it builds up, it will tend to be driven down the pathway. However, hexokinase is not the committed step to glycolysis because glucose 6-phosphate can also enter glycogen synthesis or the pentose phosphate pathway. In contrast, phosphofructokinase 1 is not only a regulated step in glycolysis, but it is the committed step because fructose 1,6-bisphosphate cannot enter into glycogen synthesis or pentose phosphate. It can only go down into glycolysis. Finally, pyruvate kinase is also a step which involves a drop in energy, and so it is irreversible, which means that pyruvate will not be able to just flow back into glycolysis, but it must go into a different pathway. One of those alternative pathways is the citric acid cycle. In fact, in the activity in Cougar, I will describe how pyruvate can enter the citric acid cycle, and this links glycolysis to the citric acid cycle. Briefly, pyruvate dehydrogenase grabs pyruvate and removes a hydrogen, producing carbon dioxide to power the acetylation of coenzyme A, which can then be bound by citrate synthase in order to donate its acetyl group to oxaloacetate to make citric acid. Again, we'll talk about that in Cougar. Today I want to talk about a different pathway, and that is where instead of going into the citric acid cycle, pyruvate goes into gluconeogenesis. Whereas glycolysis oxidizes glucose to pyruvate to make ATP, gluconeogenesis takes pyruvate and rebuilds glucose at the expense of ATP. Accordingly, each of those negative energy steps that was regulatory in glycolysis, shown in red, must be reversed by a different negative energy step, shown in green, in gluconeogenesis. For example, pyruvate kinase in gluconeogenesis must be reversed by an alternative pathway which begins with pyruvate carboxylase. Interpret the name to give me some idea what it does. If pyruvate has 3 carbon atoms in it, how many atoms will there be in oxaloacetate? Pyruvate carboxylase acts on the substrate pyruvate and it adds or links a carboxylic acid to it, so that does add a carbon atom to the 3-carbon pyruvate to make a 4-carbon atom oxaloacetate. The next enzyme in this pathway, phosphoenol pyruvate carboxykinase, is going to form phosphoenol pyruvate by burning the carboxylic acid off of oxaloacetate to power the phosphorylation so that we have phosphoenol pyruvate, a 3-carbon intermediate with a phosphate group. As the concentration of phosphoenol pyruvate builds up, and assuming the glycolytic enzymes in red are inhibited, pyruvate kinase, phosphofructokinase, and hexokinase, what then will happen to the concentration of 2-phosphoglycerate, 3-phosphoglycerate, 1,3-bisphosphoglycerate, glyceraldehyde 3-phosphate, and fructose 1,6-bisphosphate? Please pause and answer. This is a critical question. These are all reversible steps. All these intermediates will just flow back and forth in equilibrium such that all of these intermediates will be in high concentration, including fructose 1,6-bisphosphate. And of course, now we're at that critical next regulated step. At this regulated step, if the green gluconeogenic enzymes are inhibited, and if the red glycolytic enzymes are activated, phosphofructokinase 1 will add a phosphate to fructose 6-phosphate to make fructose 1,6-bisphosphate. Since, however, we're looking at gluconeogenesis, and we can assume the red glycolytic enzymes are inhibited and the green gluconeogenic enzymes are activated, fructose 1,6-bisphosphatase will act upon fructose 1,6-bisphosphate, and it will cleave off the phosphate to convert fructose 1,6-bisphosphate to fructose 6-phosphate. Again, fructose 6-phosphate can flow back and forth with glucose 6-phosphate, and for that matter with glucose 1-phosphate. Regardless, as glucose 6-phosphate builds up, it can be acted upon by glucose 6-phosphatase, and that's an enzyme again that we described back in the glycolysis talk. Glucose 6-phosphatase, which will break off the phosphate to regenerate glucose so that a liver cell could export glucose made from gluconeogenesis. Before I look at a little more detail at a couple of these regulatory enzymes in gluconeogenesis, I thought I would look at some numbers. How many ATP do we get if you burn 1 glucose to pyruvate? And then the other question would be, if we take 2 pyruvate molecules to regenerate glucose, how many ATP does it cost? And I have to point out that there's always a cost of doing business. Hopefully you remember that we get a net of 2 ATP per glycolysis. How about gluconeogenesis? To be clear, any business knows that you never get back what you put in. There's always a cost to business. So if you're going to get ATP from glycolysis, to go back and rebuild glucose will cost more than a net of 2. And in fact, if we take a look at it, Here I'm adding the ATP to the equation, hexokinase -1, phosphofructokinase -1, phosphoglycerate kinase +1 times 2, pyruvate kinase +1 times 2. So as I mentioned, glycolysis gives us a net of 2 ATP. Now if we look at gluconeogenesis, pyruvate carboxylase costs 1 ATP times 2. Phospholynyl pyruvate carboxykinase costs 1 times 2. Phosphoglycerate kinase costs 1 times 2. That's 1, 2, 3, 4, 5, 6. So whereas we can get 2 ATP from glycolysis, if we're going to be running a cycle, which our bodies do, you're going to be costing 6 ATP every time you make 2. So there's a loss of 4 in order to be using this type of a cycle. Now I would like to look just a little bit at regulation. To be clear, if you're going to activate glycolysis, you will inhibit gluconeogenesis. If you're going to activate gluconeogenesis, you're going to inhibit glycolysis. Because if you are running both cycles at the same time, you're going to be burning and burning and burning ATP, making a bunch of heat, but you won't be getting any ATP. This is called a futile cycle, and that's a concept which will be coming back again this year. I'd like to try a simple example of regulation. Here I'm showing glycolysis to the left, gluconeogenesis to the right, and I'm showing the key regulated enzymes, phosphofructokinase and pyruvate kinase. Each of these enzymes is allosterically regulated. Allosteric means other site. For example, ATP binds to phosphofructokinase in the active site and is a substrate. But in addition, there's an allosteric different site on phosphofructokinase that binds to ATP, and the binding to the allosteric site induces a conformational change that affects the activity of phosphofructokinase. Now, based upon your understanding of glycolysis and gluconeogenesis, can you tell me whether or not ATP will stimulate phosphofructokinase activity or will it inhibit phosphofructokinase activity? The same for AMP, the same for ATP's effect on pyruvate kinase, ADP activity on phospholynyl pyruvate carboxykinase, and on pyruvate carboxylase activity, and the effect of AMP allosterically regulating fructose 1,6-bisphosphatase activity. Spend a couple minutes thinking about this. If you figure it out, you're going to be in good shape for when we get to diabetes. But if you're finding yourself getting bogged down, on the next slide I have some clues. Most cells need to regulate their own internal energy even independently of what's going on outside of the cell. And what is the purpose of glycolysis? It is to make energy in the form of ATP. Therefore, how do you think an excess of ATP will affect the activity of phosphofructokinase? Based on that, again, try to answer as many of these as you can. Nonetheless, I'm going to move on. In any cell, if you have excess ATP, that will be a signal that, hey, we don't want to run phosphofructokinase because we already have a lot of ATP. Why would we make more? In fact, you can have too much of a good thing. In contrast, if you have a whole bunch of AMP, that's a signal that you're really low on energy. So AMP binds to an allosteric site on phosphofructokinase, and what does it do? It will upregulate it. AMP says we need ATP, so it will upregulate the committed key regulatory enzyme for glycolysis. How about pyruvate kinase? Pyruvate kinase is the last step in glycolysis. It is the enzyme that actually forms ATP. So if we have an excessive amount of ATP, we do not want to run pyruvate kinase to make more ATP. High energy will downregulate glycolysis, and low energy will stimulate glycolysis. Now, in gluconeogenesis within a liver cell, I'm only showing AMP and ADP. How do you think they will affect gluconeogenesis? Please pause and answer. Well, you really can't make glucose unless you have energy. If you have high ATP in the liver cell, you will be able to run gluconeogenesis. If AMP activates gluconeogenesis and it also activates glycolysis, you're running a futile cycle. So just as AMP stimulates glycolysis, it will downregulate gluconeogenesis. How about ADP? Well, you're saying that's between ATP and AMP. And it is. AMP is a sign that, wow, we are really low on energy. But ADP is also a signal that energy is low. So accordingly, ADP is a sign of low energy. In low energy, you will have trouble running gluconeogenesis, so that will downregulate pyruvate carboxylase and phosphoenolpyruvate carboxykinase to slow down gluconeogenesis. I think that when I describe enzymes purely based on language, it might be hard to remember them, whereas when I describe them with language and also include showing you structures that tie in with the language, it might make it easier. In fact, I want to take a look at pyruvate carboxylase. That was the first enzyme in gluconeogenesis. And I want to look at the structures of the substrates. I've pulled this figure from the gluconeogenesis chapter in the Lippincott textbook, and I noticed they use a different style of figure from what I use, and I thought I should interpret some of their structures. And I'd like to begin with pyruvate itself, since pyruvate will be the substrate of pyruvate carboxylase. And I'm not crazy about their figures, but since you may be reading their book, I wanted to point out that if you look at this figure where they actually show every carbon and every hydrogen, certainly that's more complicated than the way I show it. Nonetheless, you do have the carboxylic acid group, you have the ketone, you have the methyl group. And here again, the carboxylic acid, the ketone, and the methyl group. Since pyruvate is going to form a carbon-to-carbon bond with a carboxylic acid group, that carboxylic acid group has to be in an activated form. You can't just grab carbon dioxide or carbonic acid. And in particular, that carboxylic acid group is activated by linking it to biotin, one of the B vitamins. Once you've added the fourth carbon to the 3 carbons of pyruvate, you form oxaloacetate. And this is how they show it. Again, I think that this figure is a little easier to interpret. In fact, at this point, I am going to switch to the simpler figures which I prefer so that I can clarify how pyruvate carboxylase transfers a carboxylic acid group from carboxybiotin to pyruvate to make oxaloacetate. That is, the oxalic group shown in black came from the pyruvate molecule, and the acetate group shown in green was transferred from carboxybiotin. Why is the part of it that corresponds to pyruvate called oxalo? That's because it is similar to oxalic acid, which is just like pyruvate, except it has an extra oxygen atom, and the physicians will talk about the role that oxalic acid plays in kidney stones. Regardless, oxaloacetate then can be reduced to malate. You can tell it's a reduction because you have NADH being converted to NAD+. So this is named for the reverse reaction, malate dehydrogenase, so that the malate can be exported, and then the malate can be oxidized again right back to oxaloacetate. So why is it that the first step in gluconeogenesis is in the mitochondrial matrix and the rest of gluconeogenesis happens in the cytosol? Please take a stab at that. The answer is fairly simple. The first step in gluconeogenesis produces oxaloacetate, which is a really important citric acid cycle intermediate. You want oxaloacetate in the mitochondrial to run the citric acid cycle. For that matter, you want the substrate pyruvate in the mitochondria. It's the product of glycolysis, but it's pumped into the mitochondria to be used as a precursor to drive the citric acid cycle, or in this case, to replenish citric acid cycle intermediates, such as oxaloacetate. So not only is oxaloacetate a citric acid cycle intermediate, but also it can be exported from the mitochondria and used as a precursor in gluconeogenesis. So now the second part of the question, why do the rest of the steps in gluconeogenesis take place in the cytosol? Most of the enzymes in gluconeogenesis are the same as the enzymes used in glycolysis for all those reversible steps. Since glycolysis occurs in the cytosol, most of gluconeogenesis also occurs in the cytosol. For that matter, the final product of gluconeogenesis is what? Glucose 6-phosphate. And where do you want glucose 6-phosphate? Nice to have it in the cytosol where it could go back into glycolysis or into glycogen synthesis or into the pentose phosphate pathway. Or if you're in a liver cell, you could dephosphorylate glucose 6-phosphate so that you could export glucose to control blood sugar level. So I hope you see that if you understand the citric acid cycle and glycolysis, it makes perfect sense that only the first step in gluconeogenesis occurs in the mitochondrial matrix. Now I'm zooming back out to put this in the context of glycolysis and gluconeogenesis. As I mentioned, pyruvate carboxylase is in the mitochondrial matrix where it adds an acetate to the 3-carbon molecule pyruvate to make the 4-carbon ketoacid oxaloacetate, which is then exported from the mitochondria before phosphoenol pyruvate carboxykinase converts it to phosphoenol pyruvate, which then can flow freely through all these glycolytic enzymes all the way back to fructose 1,6-bisphosphate, which, if phosphofructokinase 1 is inactivated and fructose 1,6-bisphosphatase is activated, it will be dephosphorylated to fructose 6-phosphate, which can isomerize to glucose 6-phosphate, and in the case of liver cells, glucose 6-phosphatase can break the phosphate off it to make glucose and then export it. And that's as far as I want to go today with this talk of gluconeogenesis. If you have any questions, I always enjoy hearing from you, whether it's through email, phone, Zoom, or when we're in our offices again, office visits. Please stay in touch. Thanks.